Hi students, so today we're going to be talking about cellular respiration and specifically getting into the details of the phases of cellular respiration. And so there are three main stages to cellular respiration. The first one is a process called glycolysis, where we break apart glucose. And then we have an intermediate step, which doesn't really get a name, where we oxidize pyruvic acid into acetyl coenzyme A. And then acetyl coenzyme A enters a cycle called the Krebs cycle, where it's going to create a whole bunch of high energy electrons that are going to be taken to the final step, the third step, which is called the electron transport chain, or its oxidative phosphorylation via chemiosmosis at the electron transport chain. And by the time we get done with today's lecture, those words will actually make sense to you. So we'll talk about those in just a little bit. But you can see as we move through this section that we start with glycolysis, where glucose gets turned into pyruvate, and that generates a little bit of ATP. We also have in the citric acid cycle, which is the other name for the Krebs cycle, we have a little generation of ATP. And then finally, during oxidative phosphorylation, you can see that the ATP is bigger here because we're making more ATP during that particular phase. The overall equation for cellular respiration is C6H12O6, that's glucose, plus oxygen. We know we breathe in oxygen and we need it. This is where we need it. This is how our body uses it. So we're going to breathe in oxygen and use it. Makes 6 CO2s and 6 H2Os. And so the H2O water and the CO2, the water just stays in our body because it's water, and the CO2 is given off as waste into the environment. So we're going to look at each one of these phases and kind of break it down into what specifically is happening in each of those phases. And so we're going to look at them one at a time. So beginning with glycolysis, we're going to see how glucose gets broken into two G3P molecules. So glycolysis is in two phases. The first phase is called the preparation or the investment phase, and then the second phase is called the payoff phase. And so in the first phase of glycolysis, glucose is broken into two G3Ps, and this requires two ATPs. This is a lot of words that I'm throwing at you. G3Ps and ATPs and glucose and all of these things. So we're going to be drawing each of these stages together, and I really want you to be drawing. I want you to keep these drawings together, and we're going to compile them into one big drawing in class together. And so each one of these drawings is going to be important as we go through the process. In our prep phase or our investment phase, glucose gets broken into two G3P molecules. So we'll start with glucose at the top. So glucose up here, and in its preparation, it gets split into two G3P molecules. If you remember during photosynthesis, each time the Calvin cycle turned, a G3P popped out. And now the very first thing we do in cellular respiration is break glucose apart into two G3Ps. And so what we're going to be noticing over and over in this chapter is that cellular respiration is the opposite of what's happening in photosynthesis. So our two G3Ps break apart. When they break apart, they use two ATPs become two ADPs. The very first thing we do is we're using ATP, which is kind of funny because the whole point of cellular respiration is to make ATP. And so of course we start by using it, right? It's like that adage, you got to spend money to make money. So in order to kick off this whole ATP production thing, we've got to use a few ATPs to begin with. Our G3Ps are then converted into a product called pyruvic acid or pyruvate. Now you can see I wrote pyruvate up here. Pyruvic acid is in the notes. Pyruvate and pyruvic acid are the same thing. You should write that down somewhere. All throughout this process, you're going to hear words like citrate and citric acid. Those are the same thing. Pyruvate and pyruvic acid. Those are the same thing. So we're going to hear these terms, and depending on what book you read, which diagram you look at, who is the author, you'll see them referred to by both names. And so I will be referring to them by both names to do you a favor in the future as you're going through more books and more biology in the future, so that you remember pyruvate, pyruvic acid, really just two names for the same molecule. In this payoff phase, we regenerate 2 ATPs for each side. So what we do here is in our first phase, in our prep phase, we use 2 ATP. In our payoff phase, we make 4 ATP. And so what we say is we net 2 ATP. So when we account for the numbers that we lost, altogether we gained 2 ATP. There's another thing that's happening all throughout cellular respiration. We're going to be keeping track of our ATPs, and we're also going to be keeping track of our electron carriers. Remember, in photosynthesis, we had NADPH. In glycolysis, in cellular respiration, we have NAD, no P. Remember the P for photosynthesis, so no P here because this is cellular respiration. So in our payoff phase, we also have NAD becoming NADH, just one on each side. We call that NAD+, that's just its charge. So altogether, we have 2 NADH molecules that are ready to go to the electron transport chain, which we'll talk about at the very end. And we have 2 ATP molecules that are ready to go be used in the body somewhere. So these ones are done. They get to leave. These 2 we're going to keep track of as we continue. So at the end of glycolysis, we have 2 pyruvates, 2 NADHs, and 2 ATPs, 2, 2, 2. So the 2 pyruvates are going to keep going. The 2 NADHs head off to the electron transport chain. And the 2 ATPs are all done. They get to leave. They get to go be used somewhere in the body. Because remember, the whole point of cellular respiration is producing ATP energy. So we're going to take a quick look here and remember Leo the lion. Remember, Leo the lion says, grr, right? And that's how we're going to remember that when we lose electrons, it's oxidation. When we gain electrons, it's reduction. And so what we're looking at is kind of a zoom in here of what's happening as G3P becomes pyruvate or becomes pyruvic acid. And so what we're going to be seeing all throughout this chapter is this straight arrow down that's indicating that G3P, the molecule, is becoming pyruvate. But we're also going to see these little curvy arrows along the side. This means that something else is happening at the same time. So in this case, NAD plus is becoming NADH. NAD is gaining an electron to become NADH. So this is reduced. Meanwhile, G3P is losing electrons to become pyruvate. So G3P is being oxidized. So G3P is being oxidized while NAD is getting reduced. So NAD and FAD get reduced all cellular respiration long until they reach the electron transport chain at the end. So we're going to be following these electrons. These electrons eventually make all the ATP. So we want to follow these electrons. So every time you see one of these NADs or FADs become NADH or FADH2, that is where our ATP is eventually really going to come from at the end of this whole story. So don't forget to pay attention to your redox reactions as we're going through the story of cellular respiration. So when we left off, we had just created pyruvate from glucose. So we've broken glucose down into 2 pyruvate molecules. And now these pyruvate molecules are going to get ready to enter the Krebs cycle or the citric acid cycle. So each pyruvate needs to come, first of all, into the mitochondria. It's important to note here that glycolysis, the part we just talked about, that first step of cellular respiration, happens in the cytosol. It happens just in the regular cytoplasm of the cell. In order for the rest of cellular respiration to take place, pyruvate needs to come across the mitochondria's membrane into the mitochondria and get put into the proper position to enter into the Krebs cycle. So we call this kind of transition of pyruvate to what needs to go into the Krebs cycle, acetyl coenzyme A. We call that the transition step. So each pyruvate is now going to move into the mitochondria. So this is the phospholipid bilayer of the mitochondria. And then inside, we have the cristae membrane of the mitochondria. And the pyruvate needs to come all the way in. So it's going to come into the mitochondria. Each pyruvate is going to come into the mitochondria, and each pyruvate needs to turn into a molecule known as acetyl coenzyme A. Acetyl co-A is what we call it for short. So it's going to become acetyl coenzyme A. So each one does. So we've got acetyl coenzyme A and acetyl coenzyme A, and as they come in, we're also going to reduce NAD to NADH. So for each pyruvate that comes through the membrane, one NADH is reduced. And remember, these NADHs are going to go to the electron transport chain. That's where they're headed next. Also, as this happens, we're going to lose a carbon. So every time we lose a carbon, we're making carbon dioxide, and these carbon dioxides are going to leave, and they're going to leave the cell altogether. So if you recall, glucose had 6 carbons, and pyruvate has 3, but it just lost 1. So guess what? That means that acetyl coenzyme A has only 2 carbons left. So once acetyl coenzyme A has made it into the cell, it is now ready to enter the citric acid cycle, or the Krebs cycle. Those are, just like we said pyruvate can be pyruvic acid, the citric acid cycle can be called the Krebs cycle, and the Krebs cycle is the citric acid cycle. Those are the same term. They're just named, they're just different names for the exact same process inside of the cell. So at this point, what we have done is we have taken glucose and we have turned it into 2 pyruvate molecules. And remember, we did that in the cytosol. And then in our transition step, we moved the pyruvate into the mitochondria, and we changed the pyruvate into acetyl coenzyme A. And now we're ready to enter the citric acid cycle. So the citric acid cycle admittedly looks very complicated, but we are going to learn just a few things about it. So you can see at the top of the diagram, it shows pyruvic acid, which remember is the same thing as pyruvate, becoming acetyl coenzyme A. So it actually starts with the transition step in this particular diagram. And then we see acetyl coenzyme A combining with this 4-carbon molecule here, which is called oxaloacetate. Its term is right here, oxaloacetate, and it's abbreviated OAA. And officially, just like with 3-phosphoglycerate from the photosynthesis, I'm not going to ask you the name of oxaloacetate, but just like in the Calvin cycle, it's difficult to talk about this without naming them. So we're going to say that acetyl coenzyme A is going to combine with oxaloacetate, or OAA. So acetyl coenzyme A is coming in, and it is combining with a molecule called OAA, and we'll just call it that, OAA. And the very first thing that happens in this cycle is that citric acid is formed. And this is why we call it the citric acid cycle. The reason we call it the Krebs cycle is named after the scientist who discovered it. So you can see that both Krebs and citric acid make sense as the name of the cycle. And so depending on the book, the publisher, the author, and the teacher that you have, they will call it different names. Because I'm preparing you for a world of biology, I call it by both names. So the citric acid or the Krebs cycle happens next. So in the citric acid cycle, we're going to combine oxaloacetate and acetyl-coenzyme A, and we're going to make citric acid. And then we're just going to go through a cycle where we make oxaloacetate again. So remember, acetyl-coenzyme A has 2 carbons, and oxaloacetate has 4, which means that citrate then would have 6, right? 4 plus 2 gives me 6 carbons. 1, 2, 3, 4, 5, 6 carbons. Okay, so this is citric acid. And just kind of an interesting side note, citric acid is vitamin C. Now, as citric acid turns into oxaloacetate, it is going to lose carbon as CO2. So these 2 carbons are going to get to leave. And so we will have gotten rid of 1, 2, and we'll be back to 4 carbons of oxaloacetate. In the meantime, we're also going to be reducing NAD to NADH in 3 places. So we get 3 NADHs produced from this. And in what feels like just a confusing turn, we also fill a different electron carrier called FAD, becomes FADH2. And the very last thing we do is 1 ADP, 1 ATP. So after all of this, let's do our totals. So what do we make in the citric acid cycle? Well, we make 2 carbon dioxide. 1, 2. We make 1, 2, 3 NADHs. We make 1 FADH2. And we make 1 ATP. This is for 1 turn of the citric acid cycle. But just like the Calvin cycle, remember we had 2 acetyl-coenzyme A's. So we actually go through the citric acid cycle 2 times for a total of 4 CO2s, 6 NADHs, 2 FADH2s, and 2 ATPs. And these numbers are per glucose, where these numbers are per turn. So on the test, there will definitely be questions about the numbers per turn and the numbers per glucose. So it'd be a really smart idea to take down all of these values. So at this point, I hope you're thinking, well, what is the point of all of this? Because that's what I'm thinking. So let's go back over what we've made so far during all of this process. During glycolysis, we made 2 ATPs and 2 NADHs. In the transition, we made 2 more NADHs and 2 CO2s. In the Krebs cycle, 6 NADHs, 2 FADH2s, 4 CO2s, and 2 ATPs. So the whole point of all of this work is to make ATP, and so far we have made a measly 4 ATP, that's it. That's all we have. So what was the point, right? We took this whole glucose molecule and all we've made so far is 4 ATPs. We have some stuff that's left as waste, okay? What has gone as waste? These guys. Those guys are being breathed out. Meanwhile, all of these, approximately 10 NADHs and 2 FADH2s, these guys aren't done yet. These guys are on their way to the electron transport chain to make lots of ATP. So that's where we're going with this. All of these things, the CO2s, the ATPs, we're done with those. We set those aside, okay? We don't need those anymore. But the NADs and the FADs, they're still moving. They're still taking those high energy electrons and they're going to take them over to the electron transport chain where they're going to do something called chemiosmosis. And they're going to do oxidative phosphorylation by the process of chemiosmosis. And so we're going to get some really big terms in just a second. But all of the electrons from glycolysis and from the Krebs cycle and from the transition, all of those electrons are now moving to the electron transport chain, which is the big ATP making portion of cellular respiration. And this is where I'm going to leave you guys for today, and we're going to pick up the electron transport chain tomorrow.